Okay, I'm going to show you how this is done because I believe the virtual lab does not show it as well as I would like it. So we have a normal plant that's given sun. This is the cDNA. Now on the microarray, you notice that the complementary strand is a whole lot shorter and it's only 4 bases long. Meaning that the cDNA, even though it is about 6 base pairs long, only 4 of these bases need to match perfectly to what's found on the microarray. So again, normally if we're doing this in real life, we'll just take all the cDNA and then we'll put it on the board and it'll magically stick. But I want to show you the process so it makes sense. So for instance, if I take this first cDNA on here, and I'm trying to see if it matches at all, at least 4 base pairs. My question to you is, would this stick to the board? If you said no, that is correct. This one is not going to stick to this gene. But it doesn't mean it's going to not stick to the other one. So you kind of have to, you have to go through all the rest of the genes. Would this one stick? The answer is no. Going down to gene 3, would this one stick at all perhaps? No. Okay, so I'm going to finish that one off. But here is another cDNA from the normal plant given sun. Would this one fit? Well, maybe, because we have the G with a C, but A does not base pair out with G. So that's not going to work, even if I do move it. Will it fit here? Well, A does base pair with T, but T doesn't go with T, and neither would T with G. So that's not going to work at all. Then down here, wait a minute. It looks like something might work. So A does go to T, A does go to T, C with G, and G with C. Even though we are not using the C and A in the complementary DNA, that's fine. Because for this one, only 4 of them need to match up perfectly. And this one does. So in this microarray, or for this gene, this cDNA is going to stick to this one. And it'll just sit there. So I have completely attached all the cDNAs to the microarrays. So as you can tell, only one cDNA washed out. The other ones were able to stick. So I want to go through each gene and show you how they sticked. So this one we have those 4 lining up. And then we have this gene down here. I know it's hard to see because my handwriting I kind of messed up with the A. We have the T with the A, and then the 3 thymines on top with the 3 adenines, on the bottom. Nothing here. This one down here, they all matched up. And then nothing, nothing, nothing. This one was kind of easy to match up because you can see it right there. Nothing, nothing, nothing. And this one was kind of cool because you can have more than one cDNA attached to the actual microarray. So all 3 of these cDNAs were able to stick to this microarray right here. So I went ahead and did the cDNA in correspondence to the plant that was grown in the dark. So I'm going to go over it. So if you see in this one, over here, this gene, this cDNA did correctly match to at least 4 base pairs to the DNA that was on the microarray. If you can look, notice that there is a normal green one with that one. And then we have this cDNA, which correctly matched to this DNA on the microarray chart. And then we have this gene down here, in which it correctly matched. This one was super easy. That's it. There we go. Taking forever. There we go. And then over here, we have this one. I'll move that one out of the way so you can see. This one correctly matches. And this one also has a green one as well. And then we have this cDNA, which correctly matches to this gene right here. Okay, so if you notice, I'm gonna have you guys to kind of predict before I actually show you. So I highly recommend that you pause the video after I ask you the question. So here is our marker, or not marker, our microarray. Remember the three colors? We have black, we have green, we have red, and we have yellow. Remember, if they're black, it means that both sets of DNA is not turned on. If it's yellow, that means it's equally expressed in both sets of cells. If it's red, that means it's overly expressed in the cancer cells or the plant that was grown in the dark, because that was the conditions for that. If it was green, then that means it is working normally or expressed equally, not expressed equally, but normally expressed in normal cells or the plant that was grown in the sunlight. So for this one, for this gene, what color do you think the microarray would be fluorescently shined? What color would it be? For this one, what color would it be? For this one, what color? This one, what color? This one has that one. What color will it fluorescently shine? This one has both. So what color will that one turn out to be? What color? What color? And then this one, we have that one. This one, we have this one. And this one, this one. This one, remember it had 3 greens. And then this one has nothing. And then this one has the red cDNA. So, predict what color this microarray chart will fluorescently shine due to the cDNAs that were popped up on here. Pause the video.
A few moments later.
So here's how the microarray will shine given the fluorescent light. How well did you do? So let's go over it. So starting on this side, this gene contained no cDNAs. So we know that the plant with light and the plant with no light does not make this gene. Same thing for this gene. For this gene, it fluorescently shined green. So that means in the plant that was given the sun, this gene is expressed normally. Same thing with this gene, because it was also green, and there's only one cDNA that popped up to it. Now in the next one, this one right here, it fluorescently shined red because of this one cDNA that was over here that corresponded to the plant that was not given sunlight. So this means for this particular gene, whatever it represents, it is overly expressed in that plant without no sun. It's not a good thing. In this one, there is one green cDNA and one red cDNA, which then, given the fluorescent light, is then going to shine yellow, meaning that in the plant with light and the plant with no light, they were expressed equally and that they are working exactly the same. This gene, not expressed. This gene, not expressed. Moving over here, black, gene, not expressed. In this one, we have one red cDNA and one green cDNA from the plant given light, which then would make the cDNA for that gene pop up yellow, meaning that for the plant with light and the plant without light, they will equally express. So that gene is not of interest. Moving on, we are given to this gene right here. There is a cDNA from the plant with no light, and that is it. So hence it is going to fluorescently shine red, meaning that this gene is overly expressed in the plant with no light. So this will be a gene of concern and to look at to see how this alters the plant's functionality in regards to no sunlight. Black, no gene, not expressed. Black, not expressed. And then this one right here that contains 3 cDNAs from the plant that was given sunlight. So that means that in this gene, whatever it corresponds to, it is expressed as normal. And then again, this one right here, black, meaning that it is not expressed in both plants. And then this one right here, I did not do, but if I were to do, it would be colored red because there is 1 cDNA from the plant that was given no light, and so this plant, this gene is overly expressed in the plant that was given no light, and again, it would be a gene to look at. So looking at this, if I were to remove all of this so it's a little more clear, there are exactly 1, 2, and 3 genes that we want to look at in regards to this plant that was given no sun to see exactly how that plant is behaving due to no sun, because obviously in those genes are overly expressed. If this was cancer, we would want to look at these genes in regard to cancer to see why it's overly expressed in that individual and see what we can do to fix it, look at personalized medicines to see if we can fix these overly expressed genes. That was a lot to get through, but I hope you did well. We did it! I hope it made sense with this video and the resources that I have for your 3.1 day 3 stuff. Hopefully Microarray will make a whole lot more sense. So in this week, you're going to learn more about the science behind microarray technology. You will perform your own simulated DNA microarray experiment to study the expression in healthy lung tissue and in cancerous lung tissue, and then you should be able to analyze DNA microarray results, which was a lab we would have done in class if we had school. So I hope this was informative. The best thing about this video is you can watch it over and over and over again. See you guys. Bye.